say evolution, what comes to mind? Maybe, like many others, you think of the common and somewhat inflammatory notion that humans came from monkeys. As I'm sure you can imagine, this is a dramatic oversimplification of the process that provides the foundation for many biology courses. Evolution is the complex process that describes how all living things have changed since our first days on Earth. And evidence for evolution exists in spades at the microscopic level. One great example of this is how similar the cells of all complex organisms like animals are. They all contain the same basic tools such as mitochondria and nuclei that are necessary to sustain life. Another piece of evidence supporting evolution is the genetics relatedness among all different branches of life. Sequences of DNA are remarkably similar across species, even in species that you wouldn't expect, like baker's yeast in humans. At the end of the day, why should we care about this process? Evolution takes a long time to happen, and you might expect that any consequences of evolution happening now would be far removed from our lifetimes. And if evolution only happened in large multicellular organisms like animals, you would be correct. The way in which populations of cows, for example, are changing would be mostly irrelevant to our lifetimes, because cow evolution involves the concerted action of changes in millions of different cell types, and these changes would have to be passed down to new generations of cows. The problem is that smaller, simpler organisms can also evolve. Namely, disease-causing or pathogenic bacteria are evolving at a rate that is currently outpacing the development of new antibiotics. Unlike cows, bacteria are not multicellular and they reproduce much more quickly, and this allows any mutation to spread through populations of bacteria very rapidly. This phenomenon is making it more difficult than ever to treat simple bacterial infections, and today I'm going to provide a deeper explanation of how genetic information can change and how these changes eventually lead to evolution, and how that has contributed to the development of antibiotic-resistant bacteria. To begin understanding these complex processes, we must understand the fundamental molecules of our cells that change to allow evolution to happen. Those molecules are called DNA. The building blocks of DNA are called nucleotides. There are 4 nucleotides: adenine, guanine, cytosine, and thymine. Different combinations of these 4 building blocks form genes, which comprise the functional unit of DNA. Genes contain the instructions for a cell to carry out different tasks. If we think of nucleotides as words, then genes would be the sentences giving instructions to the rest of the cell. Genes are the instruction manual that contain all of the information necessary to execute life-sustaining processes. The information stored in DNA is remarkably stable. Despite this stability, accidents still happen all the time. DNA in cells can be damaged by different environmental factors like exposure to UV radiation or cigarette smoke. DNA damage typically consists of some type of chemical alteration to the individual nucleotides, and more severe types of damage can cause breaks in the backbone of DNA. These are more difficult to repair than chemical damage. Though most damage done to a cell's DNA is ultimately repaired, some types evade the DNA repair machinery in the cell. Damage makes DNA more susceptible to mutations or alterations in the sequence of nucleotides in DNA. Mutations can make it more difficult for a cell to interpret what the instructions encoded in DNA are, or they can change the meaning of those instructions altogether. Mutations do not always come from DNA damage, and instead they can come from errors during the process by which DNA copies itself to make more DNA for a new cell. Mutations sound intimidating, but they're not always harmful. Just like how misprinted instruction manuals could simply omit one word or change the order of important steps, the effects of mutations vary as well. Many mutations are neutral, meaning that they are neither advantageous nor harmful for an organism. Some mutations are even beneficial for an organism, and the accumulation of small beneficial mutations over long stretches of time is what causes living things to eventually evolve. For example, if a purple berry suddenly became toxic and one member of a colorblind species acquired a mutation that allowed it to see the color purple and avoid the berry, that organism would be better suited to survive in its environment than its peers. One key thing to understand about this process is that the effect a mutation will have is completely random. Though some types of mutations are more common than others, the gene that will be mutated is not specific. The purple-sided organism we just discussed didn't do anything special to gain that mutation, it just happened to win the genetic lottery and become better suited for its environment. So, how does an advantageous mutation get spread throughout a population? Simply put, organisms with these types of mutations are better able to survive and reproduce than their counterparts that don't have these types of mutations. There's a good chance that the organism with the new ability to see purple will pass that trait on to its children, and like their parent, the offspring with that mutation are better able to survive and reproduce, eventually spreading the advantage throughout an entire population. This is essentially how new species or groups of living things that can reproduce in nature develop and spread through the world. This phenomenon is what has been observed with the growing number of antibiotic-resistant bacteria strains in the past few years. The pathogenic bacteria are forming new strains, and a strain refers to a variant or a subtype of a species. These strains are altered in some way that makes the bacteria resistant to the antibiotics that are traditionally used to kill them. There is an endless list of ways that these bacteria can become resistant to antibiotics, so to better understand this process, I want to talk about one of the most widely discussed antibiotic-resistant bacterial strains, methicillin-resistant Staphylococcus aureus, or MRSA. MRSA is a strain of Staph aureus that has become resistant to the common antibiotic used to kill it, methicillin, over time. MRSA infections are spread through casual contact with other people, and this spread typically occurs in healthcare settings. MRSA infections are very difficult to treat and have a mortality rate of roughly 36%, and that rate is much higher than infections caused by other strains of Staph aureus that are still susceptible to antibiotics. To understand how MRSA became resistant to methicillin, we must first understand how methicillin was able to kill MRSA in the first place. Staph aureus, like many bacteria, have a cell wall that protects it from its surroundings. One type of molecule that makes up this cell wall is called peptidoglycan, and these molecules make a mesh-like structure that protects the bacteria from the environment and helps maintain its shape. Methicillin functions by interrupting the synthesis of this outer mesh, and without the mesh layer, the bacteria are not able to survive. Enough methicillin eventually kills off a population because it inhibits the synthesis of this outer layer. Now that we understand how methicillin kills Staph aureus, we can begin to understand how the bacteria evolved to become resistant to this antibiotic. As we have already discussed, mutations in DNA happen all of the time. Some of these mutations in bacteria gave them a survival advantage against methicillin by producing a new molecule that binds to and neutralizes the antibiotic so that it cannot inhibit peptidoglycan synthesis. This allows the bacteria to survive in the presence of methicillin. And because those bacteria were able to evade death by the antibiotic, they are the ones that go on to reproduce. Other bacteria that do not contain the mutation are killed off by methicillin, and this essentially selects for a new population of bacteria that has the mutation. When the new bacteria reproduce, their offspring inherit the genetic information from the parent cell, including the new mutation that causes the resistance. And enough rounds of this replication eventually gave rise to a new strain known as MRSA. This new type of bacteria is extremely dangerous because it is still able to cause disease, but now doctors are unable to use methicillin and several other antibiotics that work similarly to methicillin to treat it. This type of evolution happens all the time in nature, but antibiotic resistance has been on the rise due to human action as well. Particularly, the overprescription, inappropriate prescription, incorrect usage, and agricultural usage of antibiotics have accelerated this process. All of these actions have selected for antibiotic resistance through similar paths as outlined above. The scary thing about this process is that it never ceases, and already MRSA variants that are resistant to multiple types of antibiotics are on the rise as well. Though these variants are much more rare than single-drug resistant versions of MRSA. This new emergence is due in part to the actions of scientists and healthcare workers. Since the discovery of MRSA, there have been popular movements in healthcare to decrease antibiotic prescription. Now, if a patient does not have a diagnosed bacterial infection, it is very rare that a doctor will prescribe antibiotics. I know this can be frustrating if you are sick, but by limiting the use of antibiotics, doctors reduce the likelihood that multi-drug resistant bacteria develop and inhibit the spread of bacteria that are already antibiotic resistant. I hope that learning about this topic has demystified the broad concept of evolution and given some real-life context as to how evolution works. Understanding evolution can help combat the fear and confusion that the word frequently elicits. All living things are constantly changing, but a small subset of these changes threaten human survival. By engaging with this foundational concept, scientists have discovered strategies to cope with the fast-evolving bacteria that we see today. When the public fully understands and embraces these strategies, we are able to unite against these threats in a way that benefits everyone.